Okay, I thought this was funny when your legs don't work like they used to before. So showing the bones still present from legs, but not working for vestigial. Okay, embryonic development. So this is where we have similarities in the embryo. It's because a lot of it is a shared gene, the Hox genes that are responsible for embryo development. This is a fish, a bird, a reptile, all have similarities in their embryo. A snake, a mouse, a human, again, similarities in all of these embryos. It's because of that Hox gene. And we know that even if we modify that Hox gene, it's going to affect body part development. So they actually changed that gene and noticed that here in these pill bugs that it produced more legs actually when they did that. So you may have seen drawings like this showing embryo development. This is actually not the best depiction because these are like exaggerated because they're sketching. So again, when we're looking at this, make sure we're looking at actual pictures of embryos and not drawings like this, which can be misleading. Okay, our next source of evidence of evolution is biogeography. So this is the idea of life and its distribution of species over time and location. Okay, so species near geographic regions tend to resemble each other. But then we have continental drift and Pangea occurring and explaining why we have similarities on different continents. We have the word endemic species. This is a species found in a certain region and nowhere else. Okay, example of endemic species are like koalas on Australia. These are iguanas that are found in the Galapagos Islands. They're found there and nowhere else. Or ring-tailed lemurs on Madagascar. These are examples of endemic species. But because of that, they're located in that area, and we might have other organisms that are similar. The biogeography is also shown with our large birds. So we have similar birds, the rhea, the ostrich, and the emu, all being found in the southern hemisphere, but on different continents. So they all have very similar features, and it's thought of that they shared a common ancestor in the past when those continents were actually much closer together before Pangea and continental drift occurred, making them spread out and having their own different areas. And we can also see this by comparing the DNA sequences, and we can get a map out a tree. And we'll look at more of these phylogenetic trees next class. Okay, so movement of continental plates here shown again. Again, we had extinctions and speciations happening during this time. Okay, our last point of evidence is the fossil record. So fossils can be used to get dates and look at similarities in structures and connection between organisms and figure out common ancestors. So transitional fossils are fossils that show like in between transitional states between modern day organisms and their previous ancestors. So fossils are going to be those remains of the past. They're usually found in sedimentary rock. And we have paleontology as that study of those fossils. And they're going to show changes occurring over time. And we can use it to reconstruct. There's different layers in our rock. And we can then get our fossils from that rock. We can also get fossils found in amber or ice or tar pits as well. The issues with the fossil record is that it's incomplete. Many organisms are not preserved, and fossils are destroyed, and fossils are not yet found. So there are lots of gaps and issues with using only the fossil record as a source for as a evidence for evolution. But there are some that we can use. Yeah, so there's different also things that can happen and affect our fossils as well. We can have catastrophism when we have catastrophic events. So, or we can have gradualism, and those layers are deposited over time. Or we have different layers as well. Once we have a fossil, we can date them. There's relative dating and radiometric dating. Relative dating is we're going to assume that the fossil is similar age to that of the rock around it, which isn't always the case, but it is sometimes a good assumption to make. Radiometric dating, this is where we're going to use the half-life of naturally occurring isotopes that are present. So you might remember from physical science that half-life is the amount of time it takes for half of the sample to decay to another element. So we're going to use different isotopes depending on the year range we're in to figure out how old that fossil is. And so we can use both of these to determine the fossil age. Yeah, challenges is that organisms do not use radioisotopes that have long half-lives. Long half-lives are like uranium, rubidium. These are not elements that are normally found in living organisms, so that's an issue sometimes. Also, sedimentary rock can have compounds of different ages around them. So there's some difficulties in using this method.